This is chapter 23, the evolution of populations. We're going to talk about we're going to define populations and talk about what affects them on a genetic basis. And one of the things that is a misconception is that when we start studying evolution, it's thought that organisms evolve, and but that is not the case. What happens is populations evolve. So you have groups, and usually large groups tend to evolve over time. So there are a couple of factors that you want to keep in your mind. Is you're thinking of large numbers, and you're also thinking of a long time frame. And what we have here is just referring back to the chapter on Darwin, this idea of this evolution or this change based on natural selection. Those birds, just thinking of the birds that are most adapted for their environment, that you have a really wide range in what the beaks look like because each beak is specialized for its individual environment. And so just when we go through all of this information, think of natural selection. Think of the fact that the environment tends to dictate who is most likely to pass on their genetic information via reproduction. And there's such things as microevolution. Those can happen over generations in frequencies. And remember, an allele is a specific spot on a chromosome that codes for a protein or codes for a behavior, or codes for which a protein would lead to a behavior, could possibly lead to a behavior, or codes for a specific phenotype. I guess we can keep it pretty simple. We can think of an allele as that which codes for eye color or codes for hair color, codes for a tremendous amount more than that, but just to refresh your memory in terms of what an allele is. There are three major mechanisms that cause a change in the frequency of alleles. Natural selection, we talked about that already, and we will come back to that in the course of this conversation on this chapter, but also a couple of other new things: genetic drift and genetic flow. So genetic drift and gene flow. And what you're seeing on this particular image is that over one generation of these genes for eye color, there have been a little bit of a shift in terms of the percentage of green and brown to a 60, from an 80-20 to a 60-40. That would indicate that something actually happens. There might have been some criteria that that changed, and the thing that you don't know is if the next generation, if it'll go back to 80-20, or if it will reduce even further in terms of the ratio of green to brown eyed genes. Only natural selection causes this adaptive evolution. The idea of genetic drift, of somebody going to a new area, like a bird going to a new area, it's a 50-50 chance as to whether that bird is more adapted to that area or not. And then with regard to gene flow, that one also, the same kind of concept. It's a very hit or miss whether that particular adaptation is good for that area. Only natural selection is based on an individual's ability to reproduce and pass on their genetic information. There is a variation in traits, and that's a prerequisite for evolution. So there is a that we see that timeline, we see that step-by-step change in terms of our story of evolution. in terms of Mendel's work, remember Mendel realized that there was the randomness of sorting of the alleles. It's always a 50-50 chance with regard to the randomness of sorting. So the only thing what that's saying is it's the selection or the reproduction and the reproductive capacity of one group or one set of individuals that is actually passing this evolutionary pathway along. It's not the individual genes that are coming together. It's the fact that you have a whole group or a good portion of a species that's more adaptable to its environment and passing its genes along. In terms of genetic variation, it's it also acts on phenotypes, on what the individual organism is going to look like. So genetic variation will certainly lead to proteins being made and lead to, as I said, different behavioral aspects, but it'll also lead to different looks. Phenotype is what is expressed either visually or what we can test. And what you have are two very distinct type looking worms here. One is very much adapted to its environment in terms of camouflage, and both of them are adapted to their environment because these are moth caterpillars, and they're not only their diets are different and their environments are different, they have this capacity to actually have this movement, this kind of arching movement, which we don't necessarily associate with caterpillars. We kind of associate more of a moving along the twig, but they have this arching movement. And then you have very much a wide variety of phenotypes when we think of the fact of how many dogs there are. There are two characteristics that contribute to variation that can be discrete and also quantitative. Discrete, you can think of this as an either or, so either you have white flowers or pink ones. And quantitative, you can think of white and then everything in between till you get to pink. And so the variation can be measured, genetic variation. We can calculate it, measure it by looking at changes in nucleotides. Just to review, remember you have your DNA. Your DNA has the matching of nucleotides between adenine, thymine, guanine, and cytosine, and also in codons in groups of threes. And those repeating units can be looked at to look for variability. The idea of variability, and particularly if you have two chromosomes for each set, then you have a heterozygous set of chromosomes, measures the percent of that particular allele on that particular site, that loci, or the locus that are heterozygous in a population. So that variability measures that particular average, and it is done by comparing the DNA sequences. And I think this image, just the variety with regard to the site, the females, and the variety with regard to the flower color, in terms of the idea of this heterozygosity, remember you have the possibility of the two big B's, so you have homozygous dominant or two little c's, homozygous recessive, or you can have the heterozygous where you have the big and little. So you have this variety because of the nature of the sex chromosomes. An example with regard to geographic variation differences, by separating gene pools. So geographically, you have this mice population on this island that the populations are isolated. Due to this isolation, there is a chromosomal difference between the two populations. They started out as a very it might have been a founder mice. One mouse went to one part of the island, one group of mice went to another part. They have been interbreeding amongst themselves, and over time those two populations now have chromosomal variations. Geographically, that's geographic reason for variation. Another geographic variation, an example, that's all we're doing here is giving you two examples of geographic variation. Because of adaptation to their environment, what's happening is this fish, Mimico, the fish, I believe it is something similar to that term, have adapted to different temperatures. So this particular species has adapted to the very cold waters in Maine, all the way down to the very warm waters in Georgia. So you have this variation, and not only has are the fish found in these varying temperatures, the adaptive allele can be seen. So the allele difference between the fish can be seen. And this is because of geography has caused the various populations in those temperatures to choose natural selection, so to choose those fish that are most adaptable at that temperature. So natural selection is the reason for this variation. Mutation can do some of this. So mutation can arise. Mutations aren't always bad. Only mutations that happen in gametes can be passed on to offsprings. And so one type of mutation that we talk about are point mutations. They can vary. They can be in non-coding regions, and generally if they're in non-coding regions, meaning they're not in regions that create proteins or dictate the creation of proteins, they tend to be harmless as far as we know. And then they can also be neutral, particularly if there's already another sequence that codes for the same thing. The effects of point mutations, if they change a protein, like you're seeing over here, they can be very harmful. If a protein isn't made or the wrong protein is made, that can be harmful. But every once in a while, a mutation that makes a new protein can be beneficial, but for the most part, it's harmful. These are called point mutations. Very wild and interesting application of this idea of altering point mutation. So altering the gene number or position is with regard to chromosomal mutations can be disruptive or delete or rearrange. They're typically harmful, but duplicated genes are less harmful. And I'll start talking about this, but I'll pick it up on the next one because it's really wild. We had an ancestral odor detecting gene, and that's been duplicated over 1,000 times in humans. But in mice, it's been duplicated about 1,300. And the interesting thing is this duplication, although it's considered benign, again, we'll pick this up on the next one.